Yeah, so you do pick up, well, that's the exact question. So you find these hits with receptors, you find the concentration responsive hits. The question is it adverse or not, right? And that's a question which we've been debating for a long time. You see bioactivity, but does bioactivity translate into actually a phenotypic result? Because that's what we really care about. So what we're trying to do is say, okay, at a cell level, and this is true for actually specific and non-specific chemicals. So specific, non-specific being the ones that don't have a specific molecular target with a high potency, right? It also, I mean, you may hit a target at really, really high concentrations, but do you actually hit the target at micromolar concentrations or not? So those are actually far and few in between. You don't get really high numbers of micromolar potencies for all of the chemicals. So in those cases, and even for the specific cases, one of the pathways and the cellular systems that I actually really care about, and some subset of us are really interested in, are these adaptive stress pathways. And what are these adaptive stress? So the chemical's not going to go in and bind something directly, but what it'll do is after metabolism, it'll produce a bunch of reactive oxygen species or various reactive species. Okay, what are they going to do? They're going to increase oxidative stress. That's going to engage the oxidative stress response machinery through KEAP1 and NRF2. Unfolded proteins. I mean, you know that unfolded proteins are going to happen all the time. Whenever you have adducts binding with proteins, they're going to affect the folding machinery, and that's going to set up the unfolded protein response. And the third, of course, DNA damage response. You don't have to have, I mean, related to oxidative stress, you can form again adducts with DNA. You can get single strand breaks or double strand breaks and engage the DNA damage response system. So these systems have actually evolved. They're in all cells. They're in different species. And so people have argued that this is a really nice generalizable approach to actually look at this question of toxicity for these non-specific chemicals. And so the apparatus that cells have for picking up this stress is there's a sensor sitting around. It's a very general modular architecture that's been conserved across these different stress response pathways. There's a sensor. There's ATM and ATR for DNA damage, for unfolded protein response, it's PERK, or ATF6, IRE1 alpha. For oxidative stress, it's KEAP1. These sensors bind their transcription factor translocates the nucleus, turns on gene expression, effector molecules come in, they clean up the damage, right? And they have feedback inhibition to shut down the stress. Now, if the stress level is low, you're going to get survival. Cells are going to be proliferating or growing as it normally should. But if the stress level exceeds some threshold, you're going to switch over into a different cell fate. You're going to go into potentially, depends on how much the damage is, you could go into autophagy. The cell might say, okay, you know, I can't really survive. There's too much damage. I'm going to just start chewing up myself and eat up and use the resources. Or I'm going to go into apoptotic cell death, or even go into senescence. What we don't really understand is what are the key switches, molecular switches. There's a lot of hypotheses about what those switches are, but these transducers and these other switches, where they are and what they do, it's not really well understood. And that's part of the question that we're trying to attack in a very generalizable way, so that we don't build one bespoke model that we can never use or never adapt. So we have a lot of data resources, and you can actually access those over the CompTox dashboard. You asked about the data sets. We have a huge amount of transcriptomic data coming along, cell painting data, which is really like high content imaging, but you're familiar with like cell profiling. We have animal testing data on about 1,000 or so chemicals. And we also have other sorts of information that are freely available and can be downloaded from our website, comptox.epa.gov. All right. So the strategy at a high level for us was, let's start with a set of chemicals that we know something about, referencey chemicals. And then let's also include environmental chemicals in the mix. And let's try to assemble the information about those. There really is a reason for using a knowledge graph, and I'll go through that, even though I'm not going to dive into any applications of machine learning on that knowledge graph today. But we're trying to structure the evidence that we have about the chemicals, about the genes, and about the pathways that they perturb, be they stress response pathways or the phenotypes. And then taking that information, we construct a Boolean network, derive those from the knowledge graph based on key genes, do simulation on that and optimization to find what the phenotypes are of the theoretical model that we get out of literature, and then look at the doses of different chemicals and concentrations of different chemicals in a specific model to see how does the treatment dose correspond to the cell fate outcome using that model. Right, so again, not a very sexy approach to doing information extraction, no deep learning, no nothing, very old school. We wanted to build really a bootstrap data set first. Before we build something really fancy, we're trying to assemble in evidence about chemicals. And this paper that you're looking at work was done by Brian Chambers, who's a postdoc with me, just left late last year. And we built a pipeline to extract evidence about chemicals and their potential relationships with adaptive stress, as well as chemicals and their relationships with genes. And it's based on this notion that if something is mentioned many times in literature together, there's potential for there being an actual relationship that makes sense. And we use various statistics like pairwise mutual information or pointwise mutual information to prioritize which relationships actually make sense. This lets us avoid very spurious types of relationships that might be out there in the literature. So we have very simple information retrieval approach. We come up with sentences. We have a very simple pattern matching approach to identify what the potential assertions are that are stated in there about this chemical turns on this gene, or this gene activates this gene, or this chemical turns on this particular pathway. And we store that information so we can compute with it and analyze it. So we get a bunch of key relationships out of the literature, and these are some of the statistics on those. We've standardized them to the degree that we can compute with them. There's no very fancy thing over here. It's a very simple, a head entity, an action, and a tail entity, which are the chemicals, actions, genes, action, genes, and genes action with these cell fate or cell stress pathways. And now we can basically use that and compute with those and do something fun. So here's the critical question. Once you get a knowledge graph, and this is sort of the dream, right, that you can take this massive amount of knowledge and de novo come up with this really perfect network that describes your biology. Well, it doesn't work so simple as that. You have to use prior knowledge. So we had to go back to the 3 or 4 review articles to look at, okay, what do we know about DNA damage response and increase in autophagy? What do we know about DNA damage response and impact on cell cycle and apoptosis, and DNA damage response, and I think there's one other thing in there. No, that's it. And assemble those assertions into a model that describes how you could infer a particular gene or protein responding to an upstream signal. So you basically take these assertions about relationships between genes and downstream genes and assemble into a Boolean threshold network, where you take activation and inhibition relationships to weight the inputs, and you have a threshold, which again is something that you have to hand pick initially to come up with an outcome. Now, Boolean threshold networks are a very simple idea. You have various inputs coming in, and what you see on the bottom over here is an example trajectory in which you have time points going from the lowest row up high, and you see activation patterns propagating along this. And when you hit, so with DNA damage, after 1, 2, 3, 4, 5 steps, you hit apoptosis, right? So it's a very simple idea, but it captures and integrates a lot of complexity and also captures some feedback inhibition as well as feed-forward steps. All right, so well and good. Now what? Okay, well, so to do this, we built a little bit of a framework to analyze knowledge graphs, analyze Boolean networks. And the most important thing is, even with a smallish Boolean network of size 50 with about 150 edges, it takes a long time to exhaustively search the entire space of possibilities. So we came up with sampling strategies that can reasonably approximate the state transition graph and what the attractors should be. Then what? So here's what happened. When we actually simulate that knowledge-derived network, it doesn't actually work. It doesn't produce any of the attractors that are stable. Right? You can show a path going to apoptosis, but you don't see repeating patterns of apoptosis. It doesn't get stuck in a cycle that says apoptosis is a stable outcome necessarily. Or it doesn't give you autophagy, or it doesn't give you cell cycle outcomes. So what do you actually do? So that's where we said, okay, we have structured knowledge about the key players in this network. Let's actually try to, what you can do is you can do trial and error. And that's what most of the Boolean network approaches are You spend a lot of time reading through papers and adding relationships, removing relationships, changing your update functions, doing this by hand. In fact, I had a postdoc about 10 years ago, John Jack, who had to do this, and I think it near burnt him out. And so we didn't want to do that again. We said, okay, let's use the knowledge graph, and let's do evolutionary search to come up with an initial set of models that are plausible, and then use evolutionary search to alter the weights, add more relations, remove relations, and see if you can come up with an optimal BN that satisfies an objective function. And for us, the objective function in this particular case was, can you produce a stable state that corresponds to apoptosis? Can you produce a stable state that corresponds to cell cycle, progression, and autophagy? Very simple. And, we actually did that after like 2 or 3 generations of search, and I'm not going to go into all the details of this evolutionary, and there's many other optimization approaches available. You actually come up with this slightly modified network that has about 56 nodes, 143 relationships, and you can actually find these stable phenotypes in there. So next we brought in the transcriptomic data. How do you bring in the transcriptomic? So we've run this study on HepRG cells. We have 1,500 chemicals. About 300 of these we have evidence about actually activating DNA damage response and causing perturbations and resulting in apoptosis and autophagy potentially. So we've done concentration response modeling. So we have 22,000 genes for each chemical, and we're not going to look at all 22,000. We've analyzed each chemical that we know perturbs DDR. We've estimated their potencies for the key genes. In this particular case, 36 of those genes overlapped. So we know that the dose, the concentration at which it actually switches on. Again, this is an assumption, right? It's an assumption that even though this is bulk data, the assumption is that that particular gene is going to be turned on at this particular concentration. With that knowledge, you can actually construct a perturbation matrix in which every concentration, which genes do you think are turned on and which genes are turned off? And that's what you get shown over here in this particular graph. And now you can take each of these perturbations for all 320 or so chemicals and push them through the simulation and ask the question, which stable state, which attractor does this particular treatment lead to? And we did that. And we found that, and here's a summary of the results shown over here of a select sample of results. First thing you find is that at the low doses, almost all the chemicals at the low doses show you cell cycle is on. Now, in this particular case, these are HepRG cells. They're not particularly cycling cells, but that's where most of the evidence comes from cancer cells. So when it says cell cycle, that's normal growth potentially. So most chemicals at the low dose on this graph you can see are producing the phenotype of cell cycle. As you increase the dose for some of the chemicals, what you're seeing so the green boxes, the green circles correspond to normal function. As you increase the dose, you start seeing the orange circles, which are autophagy, and the red circles that are apoptosis. So you're seeing the critical transitions happening as you increase the dose. And a single chemical at different doses is producing different phenotypes, right? So that's where we are. And so what is our performance if you were to go back and compare it with the real data? Sensitivity is awful, but specificity is really high. Why is the sensitivity bad? Probably because the knowledge that we used over here is general knowledge about cells, various cells integrated into one place, and not really specific to HepRG cells, right? So clearly what we're seeing is that there are key perturbations that switch the DNA damage response system to different phenotypes. At least that's our result that we're finding, and we're doing further validation of this. So with that, I'm going to stop and just summarize. Of course, there's much more to do, and there's many, many caveats to this. We think Boolean network modeling and attractor landscape analysis is a valuable approach to get insights about how stresses cause these phenotypic shifts. We can generalize these BNs, so we can actually easily switch over and start developing a model for UPR. We've already done that. And other adaptive stress pathways without doing too much more manual crafting. Evolutionary computing is a really cool approach. It's actually a machine learning approach. I don't know, we don't talk about it very much, but it is machine learning. for optimizing the ends, because God knows we can't do it by hand, and there's a lot more data coming in. And last and not the least, we've run a time course single cell study to verify and validate these results at a single cell level in HEPA-RG cells with a subset of the chemicals, and hopefully we'll be looking at that data in the coming year. So I would like to thank a lot of people that did all the legwork. Weston Murdoch, who's a student, he's actually an undergrad, recent undergrad. He's doing a lot of the modeling and analysis. And then Brett Hagen, who's recently joined, who'll be taking the knowledge graph forward with various graph neural network approaches to try to see how we can expand it and how we can perhaps select the right edges to include in our models based on what's available in the graph. A lot of people at EPA that have been involved, the entire team that does transcriptomics, Josh, Logan, Joe, a bunch of others, previous postdocs, and BioSpider, who generated a lot of this data for us out of a contract with us. So with that, thank you. I'll stop there.